Okie dokie. All right. So, this picture, I hope you understand this. Each one of these blobs is a tertiary structure. There's 4 tertiary structures that come together to make hemoglobin, which is the oxygen transport pigment in your blood. Extremely cool stuff. Now, that's the quaternary structure. All right. A couple of other things I want to say about proteins is about the naming of proteins. Now, these rules aren't hard and fast, but in general, here's what happens. If you see the name of something ending in IN, so ending in IN, then it's what we call a structural protein. So, associated with the structure of bodies, of organisms. So what we can have would be, as examples of these, here's some famous ones. You may have heard of actin. You may have heard of myosin. These 2 proteins are the proteins of muscle fibers. So actin and myosin, those are classic examples. As I just mentioned, hemoglobin, that's one. Keratin, which of course, nails, hair, structural, that kind of thing. So there's a few great examples by common name of structural proteins. On the other hand, if we have proteins that end in ACE, so that ACE ending in ACE, these guys are special. Let me put an underline under that. That looks kind of crazy. So ending in ACE, these ones are equal to enzymes. Enzymes are proteins of action. Proteins that catalyze biological, and catalyze is kind of a term that means speed up. Later on, we're going to say lower activation energy. This is all for later. So enzymes that catalyze, I apologize for this little fill-in right here. That just kind of goes with the word. But things that catalyze biochemical reactions. That's what enzymes do. So, for instance, the chemical reaction of breaking a glucose off of starch. That reaction is enabled or sped up or caused to occur by the enzyme amylase. So I should throw up a couple of examples of this. Example 1, amylase, as I mentioned. Another example could be, let's see, peptidase. Peptidase is a general name, but this one has the function built in. So if ACE has to do with being an enzyme and breaking something down, then peptide refers to what it's breaking down. So peptidase is an enzyme that catalyzes breaking peptide bonds. And of course, you know what peptide bonds are. Now I want you to think, where would you expect peptidases to be active? This is a really good question. So, the radical release of peptidases occurs in your small intestine. So you've swallowed this food, and then it churns around in your stomach for a while, and then we send it down into your intestine, and your intestinal cells, well, and other structures, your whole intestinal tract is collaborating to dump these enzymes onto that food. And of course, you know, if you had a high protein meal or something, you got a lot of protein that needs to be broken down. And that's exactly what happens. And of course, a lot of that stuff is absorbed into your cells and the whole process continues. This ties in, of course, with essential versus non-essential amino acids. We've got to actually break off from our food and absorb the essential amino acids because we can't make them for ourselves. So they have to come through the diet. And all that relates to enzymes. So we eat it, chew it up, swallow it down, and then the enzymes take it from there. And the enzymes will be breaking peptide bonds, and then those amino acids are going to be absorbed. And many of those amino acids are essential. These are the ones that we need to get into us because we can't make them otherwise. So that's the big picture on proteins. Now we're going to move to the final category for our fun action, and that has to do with lipids. I'm going to start lipids today, but we're going to really get into lipids when you guys come back and watch yet another videotape on Thursday. So there's proteins. Let's do lipids. Lipids are so cool. I saved the best for last. Uh-oh. No, wait. A little bit more. I skipped something. We're going to do lipids soon enough. I skipped something with proteins. So there's a big phenomenon we have to talk about, proteins and denaturing. So this process of denaturation is unfolding or shape changing due to environment. Unfolding / change of shape, changes in shape of a protein due to the environment. What do I mean by environment? The classic things that cause denaturing. Here's the big three. So the big three causes of this phenomenon. One, heat. So cook it, change it. Everybody in this room has witnessed those changes. Think of pink flesh, a steak going onto the grill, and look what happens through the course of time. Color changes, consistency changes. So when that thing was still pink, I hope I'm not grossing anybody out, but when that thing was still pink, you know, pick up that steak and flip it around. You know, it's loose. And then let it cook, maybe overcook it, and grab that thing and pick it up, and it's like maybe a little bit of flex, but not nearly the flex that it had before. So in this particular case, the shape is changing, and actually there's a rigidity to it when it's cooked. That changes it. The second one is acid. Of course, now I've got to talk about acid again. What's acid? Remember the pH scale, where OH minus and H plus are equal, that's pH 7. And then, of course, when we increase the concentration of H plus relative to OH minus, we're in acid land. And acid land, of course, has a pH scale that goes down. So like a pH of 6, 5, 4, 3, 2, 1, those are acidic. If you get down into a pH of 3 or 4, you can just drop a steak or a piece of fish or something right into that acid, and it will denature it. It will cook it. So if you've ever had something called ceviche, that is acid cooked fish. It's awesome too. So acid, and so a good example of this is fish kind of cooked in citric acid. That's how they make it. In citric acid is ceviche. And that's a delicacy. You can go to, there's got to be hundreds of restaurants in Denver that serve ceviche. I had some in Vail like 3 weeks ago. Oh my God, it was bad. It still tasted like fish, which means that they didn't have enough acid going in that thing. Anyway, if you find a good place that knows what they're doing, give it a try. The best I've had in Denver was at a little trailer on Federal right around Alameda. Good stuff. Okay, so that's acid cooking. Heat cooking, acid cooking, and then the last one, another way to change proteins, I want you to think about this. Think bacon. Think, if you're from the South, country ham. Think beef jerky. You know, maybe not that. That's dried much more so than the other two. But salt. Salt curing is really denaturing proteins by salt. So salinity. Salinity, that is our third reason, or third causing phenomenon that denatures proteins. And it's super important. All right, so now, as promised, we can finally get to lipids. So I'll go over to this side of the board, and we can start to talk about lipids in general. And that's what we'll do. The loose definition of lipids would be hydrophobic molecules, hydrophobic substances. So these are things that hate water. Wow. I got visitors.